Each registration is unique and grants the right for the individual registrant only to view this webinar. Sharing this webinar with unregistered viewers, either during or after its performance, violates Pharmatech Associates copyright and is prohibited. So if you haven't, if you haven't already done so, please take a moment to review the disclaimer. Next. Thank you. Now I will turn over the presentation to Bikash.
Okay, thank you very much, Sylvia. Nima and I are very pleased to be here today. We're going to take you through hopefully a very interesting presentation on the application of in silico modeling, where we're seeing it really transforming CMC strategy, making profound impacts in terms of both timeline compression and really helping us accelerate and gain process insight in a way that's both cost effective and time efficient. So we'll share with you some strategies as well as some of the case studies that we have performed together to give you a sense for how it's being applied and what some of the advantages might be. So today's objective there is to kind of walk you through some of the basics of how in silico is being applied in the CMC world today, talk about some of the key benefits, what it means to you as a product developer, drug sponsor, manufacturer today, how it ties in with a regulatory strategy today. In silico is being broadly accepted, at least among stringent regulatory authorities. The value of in silico, in some cases, as a surrogate, but in many, many cases, as a complementary component to demonstrating process insight, demonstrating product insight, has been really game changing. And so we'll talk about how this can be applied today in some of the key areas of CMC, the workflows, and hopefully that will give you a much better sense for where the opportunities are within silico. And hopefully it'll also demystify a little bit of some of the uncertainty around in silico. What we're talking about today also, I want to make sure, is not AI. We're talking about modeling here. And so a lot of folks are talking about AI. AI could, of course, be folded into this, but we're talking about really digital modeling as it relates to CMC. So this slide kind of gives us the big picture. It's a map of where digital tools fit across the entire sort of drug development and manufacturing journey. You know, on your left, you see chemistry, process development, scale up, all the way to manufacturing. There, on the left side, we've got digital R&D, where we're doing molecule identification, high throughput screening, data analysis, experimental design, even early stage process design and process optimization. On the engineering side, there's front-end engineering. All of these things have a digital component that can be added to either complement, provide greater insight, or to drive speed to insight there. In the center, we've got digital design. Digital design really is sort of that bridge. It's the bridge between development and ultimately our manufacturing world. There, this is where digital tools to design and verify your control systems, your operating policies, even train operators virtually allow you to do this with some of the digital tools that are out there. And on the right, you have sort of the digital operations side. This is where manufacturing lives for folks that are looking to gain insight into the manufacturing floor, gather data there, optimize how long you can run a process, what the proper and optimal design spaces are. For folks that are looking to put in control strategies, classical control strategies in the terms of PAT and other sorts of equipment solutions. This is where the manufacturing world is. It's all underpinned with this notion of process modeling. These are foundational elements: process modeling and simulation, and then sort of digital applications on the back end. So we call this our digital taxonomy. This, you know, if you think of building a digital strategy and digital CMC, it is sort of like peeling an onion here. In the innermost layer, we've got the modeling. This is the foundational component. You're creating a mathematical or computer model of your process there, representation of the process, and trying to understand and describe the mechanisms so that you can predict the outcomes. This is where we're beginning to first build the linkages between our CPPs and our ultimate CQAs. We move to sort of the simulation world here, where we can run these computational models there over time, begin to interrogate the process design space, try and understand how it behaves as we move across different parameters. we may not be optimizing a single CQA, right? And so this is one of the biggest challenges in process development is how many runs can I make? How much will it cost? At what size? How do I manage the scale dependent, scale independent parameters? All of this can be addressed in simulation without having to run any experimental data. So it's a huge tool, huge lever that we can leverage when it comes to trying to understand what the process design space is. And then it culminates in sort of a digital twin where we build a virtual or a digital copy of the unit operations that we're ultimately trying to use at whatever scale we're interested in, but at commercial scale. And you can use this to link into your architecture if you're talking about using targeted PAT, if you're talking about simply just gathering data, SCADA data in which to understand how your process is behaving, how your equipment's behaving. This is where we start to link that with our virtual twins, and we're able to run simulations and then ultimately control the process intelligently. Nima, you want to talk about this slide?
Yes, very good. Thanks, Bikash. Good day, everyone. As Bikash mentioned, these kind of models are not data-driven models. We could have some like a hybrid modeling, but the foundation is based on the mechanistic modeling. And mechanistic models based on like a first principles are developed based on thermodynamics, physics, transport phenomena, kinetics. And because these models develop based on the science, these are like extrapolatable. So we are not data dependent. And the construction of the models start from like a material to equipment to process. So there are lots of like a scale dependent properties, like mass transfer, reaction kinetic in terms of the, when you go to a scale of what would be like the dead zone that could give us different kind of reaction profile, and versus some scale independent properties like a solubility or reaction rate that is not a scale dependent. So combining all of these together, as you saw in the previous slide, they're going to be the foundation, the core of the models. So some equations and the mathematical models that describes material properties, equipment performance, equipment characterization, and all the way to the process. The second layer would be like the simulation or running all the models together to kind of represent the process in a mathematical way and a computational way. And connecting to the SCADA system, now we would have like a two-way communication that some data could come from the SCADA and going to the models, getting some parameter updated in the models, and we can go back to the process for advanced process control. That is kind of ultimate level of the digital twin.
Yeah.
So combination of all of these would be very helpful for like a scale up, tech transfer, optimization, many different applications that we are going to show you 3 case studies today.
Right. Perfect. And I think it's the key thing I like about this is that it's iterative. We're building layers here. We're taking it in phases. We're gaining insight. And as we're doing this, at the end of the day, the goal of drug development and process development is to end up with a filing that is complete, is convincing, is defensible. There, we're gaining insight as we move along here. It allows us to do more interrogation of our process instead of running. In some cases, when we do some of these biologic processes, it's so expensive. When we try and mention DOEs, people just say, no, you can't do a DOE. So how can I gain objective insight, orthogonal insight without running a DOE? Well, I can do it in silico. Okay, Nima.
So thanks. As I already explained about the mechanistic models, so we start from like understanding the mechanism of the reactions or the transport phenomena or any interaction between the molecules or the cells. And so if you think about like a kind of like a temporal or a spatial optimization, or when you go to like a scale up, let's say like a KLA in a bioreactor or a hydrogenation or like a spray drying that we're going to have case studies today, there's no data available at the scale, at the like manufacturing scale. And as Bikash mentioned, running a DOE at manufacturing scale is kind of impossible considering the cost and the time and the amount of material is required to collect the data. That's where the statistical and data-driven models fail and cannot use those kind of models for a scale-up and the tech transfer. So now we can get the benefit of both worlds, some properties and some characteristics of the equipment or the material that could come from even the smaller scale lab data, as I mentioned, like a solubility or reaction kinetic, and then combine it with a mechanistic model that could be used for like a scale up and using this kind of hybrid model. So different approaches and a different application depending on what you want to do and what kind of question you want to answer, and also in terms of the sensitivity of the models, because there are lots of different kind of way to solve this model, and that depend on like kind of context of use, what we want to answer, how we want to answer that, and what is kind of end implications. As Bikash mentioned, when we go to like a process development all the way to the CMC, there are different questions that would be needed to be answered by for the regulatory submissions, and all could come from these approaches for like the design space development, part, nor the scale up, risk analysis, many different kind of answers that we could provide by this kind of the models.
Great.
Next slide.
Yep. So when you think about some targeted areas where you see huge benefit from using in silico, so process design, where we're doing it, we can do predictive stability. So when you think about stability, it's very difficult to get around. If you're doing a generic drug, a small molecule generic drug, you need at least 6 months real-time stability. If you're doing a new chemical entity, small molecule, you need 12 months. If you're doing a biologic, you need 12 months here. And in some cases, you have to have this data, right? And Nima mentioned this notion of context of use here, and this is a very critical principle to keep in mind. Context of use is a concept the FDA came up with, which basically says, what is the intended risk around this model being correct or wrong? And so if we're using this model, for example, to file faster, but we make a commitment to append with the actual real-time data, what is the risk of the FDA allowing you to start sooner? It's incredibly small, right? So the context of use is very different than if we were using it to do PBPK, for example, of physiological behavior in a body. Now we're trying to mimic a phase 1 clinical study, and that is a more complex undertaking. So the prerequisites, the guardrails around that sort of modeling are considerably more intensive. So understanding the context of use is a key component when you're putting your digital strategy together. We talk about scale up. Scale up is something that Nima's mentioned a couple of times here. We have scale dependent and scale independent parameters. How can we manage these scale dependent parameters in silico experimentally using first principles, or maybe first principles complemented with small scale empirical data sort of as a calibrator to try and predict the behavior and identify the optimized process design space. So as we're going through this, we are beginning to link the CPPs, the raw material, the key raw material attributes, as well as our CQAs, which is going to be the end game for your module 3. So you're really accelerating your ability to bring together all the kinds of parts of your product and process development. It's sort of a cogent and cohesive story. And so, as I said, regulators today are very comfortable with this notion here. The level to which they will allow you to lean on in silico depends on the use. Again, surprisingly, EMA is very, very pro model, however, not as a surrogate, as a complement. So they strongly encourage drug developers to use in silico modeling as part of their development team, because it gives them a greater level of confidence as to what if, because you're always going to have limited empirical data at the end of the day. So FDA will take this in some cases as a surrogate, depending on what we're trying to put forth. So the role of in silico has moved from sort of a novelty, I would say, where it was 5 to 7 years ago, to really a core component of any CMC strategy. So we're going to walk through some real world case studies here, just to give you a flavor for what we can do and give you a sense for how we attack these problems. And then hopefully you'll have some questions at the end. Nima, you want to start up with the spray dryer scale up?
Sure, very good. So 3 case studies that we are going to present for spray drying bioreactors and stability prediction. And as I mentioned, these are they have like the spatial and temporal kind of context to that. When we scale up, now we have like kind of a spatial component that what would be like a droplet generation, evaporation, particle interaction, coalescence, all of those things that would be different in the larger scale. And for like the stability modeling, now we have like a temporal component. You have to collect 2 years of the data if you go for an experimental way, or we could do like reaction mechanism and the stability prediction in silico that would help with like shortening the time for that kind of stability prediction and the filing. Starting for the spray drying is a very kind of complex process, and especially when we go to like a different product, like let's say like an inhalation product with the amorphous dispersion. Now the properties of the product that is produced from the spray drying would impact the drug delivery. If we go to like a larger particles, so the particle gonna hit the back of the throat of the patient. It doesn't go to the lungs. If we go to like very small particles, that would go to alveoli versus bronchi, and we become toxic. So you can think about changing the pump rate of the spray dryer or changing different atomization. It would change the drug delivery to the patient. That is like very, very impactful for when we predict and we understand how to control the process. Next slide, please. So what is happening during the process development and the clinical stages? Companies start from lab scale, like say Buki 290 spray dryer, and so these spray dryers using the two-fluid atomization. And you can see on the right side, that's the kind of typical particles that form from like a lab scale. And then going to like a pilot scale, something like mobile minor or a PST1. So we still have like kind of very thin shell particles, and these particles very fragile. They have different kind of dissolution and disintegration rate. So all the tablets and the formulation that developed by this product goes to the clinical trial. And the problem is when we go to manufacturing, now we go to like PST4, PST3, is a totally different atomizer, different chamber, different drying mechanism. And you can see on the lower right is the type of the particle, and the flesh or like the thickness of the shell is very, very different. Now it's going to change the drug delivery and the dissolution time. So what going to be challenging for the companies is how they can tune the process parameter, how they can select the atomizer or the process condition that provides the same disintegration time and drug release for the tablet that they already have the clinical data. Next slide, please. As I mentioned, it's a very kind of complex process, that there are lots of like scale dependent properties, and then the transport phenomenon and evaporation happens in the chamber, starting from the feed composition or the properties. What would be the impact of if you increase your HPMC or the excipient on the viscosity, surface tension, density? That would directly impact the droplet generation and eventually the particle size distribution. So atomization, or when we are selecting different cores and orifices for different atomizations, and also how we're going to run the process. Increasing the pressure, it increases the flow rate of the feed, and also decrease the droplet size distribution. Inside the chamber, so these are not like a plug flow system, so there's like a back on back flow and kind of recirculation of the material that you're going to see in the next slide. And these particles going to interact with each other, breakage, coalescence, that would happen for and kind of altering the particle size distribution in contrary to the droplet size distribution that is formed. And all the way we go to the cyclone. So some of the D10 or D5 flying out from the different cyclones, depending on the geometry and the flow rate of the gas, and that would also change the final particle size distribution of the product. Next slide, please. For all these cases, we have to simulate from the material to the particle microstructure to individual particles and eventually to the powder. And depending on what kind of question of interest or the context of use we have, some examples you can see as a CQA on the right side. Is it residual solvent content? Is it particle size distribution? Or it even could be some like a kind of key process kind of index or the performance index for the process that could be yield, right? So now we are starting from like going to the like underlying physics and the thermodynamics. What are the parameters or the stages of the process that are going to impact each of these CQAs? The fishbone diagram that you see here is developed for the residual solvent content, but it will like similar for the particle size distribution or other properties. What will the impact of the chamber size or the design or the nozzle type? You know, where is the lens located? If I bring it like 5 inches higher or lower in the chamber, it's going to impact the coalescence and the distribution of the droplets inside the chamber. So long story short, so we start developing models and the equation for each of these. That's the core part of the diagram that Vikash showed in the few last slides. Then we're running all these models together. We get to like a second layer of the digital twin that is called like the simulation. So simulation is simulating what's happening from the material to the product to the process, right? Running all of these gives us that kind of holistic approach. Next slide, please.
Yeah.
This example is for just for atomization. We you know, if we want to talk about like a spread rank, it would take like a full day. Just very quick kind of a snapshot on what is happening. As I mentioned, like atomization, now we would have different options, selecting different orifice, different cores. You know, how we run the process, you know, what pressure and what will be the material properties that going to impact the droplet size distribution. As Vikash mentioned, is a correlation of the CMA, material properties, viscosity, surface tension, density, CPP, the pressure in the atomizer, the type of the atomizer, location of the atomizer, and the CQA, which would be like particle size or residual solvent content. Now we are running at the PSD4, PSD3, which is huge. They need a lot of material. And when we come from the clinical trial or the pilot test case, there's not much material available even to do any of these things. So starting from like a developing model, so we use computational fluid dynamics to simulate the droplet generation, the evaporation, the droplets that hit each other, and we have coalescence or internal flow inside the chamber. So combination of all of these parameters could give you like 600 or 1,000 different kind of like a DOE. That is impossible to run with the experimental way, but in the simulations is very fast. We could do like very quick screening and see what will the impact of different process parameter on my CQA. Next slide, please. After atomization, now we go to the chamber, and you can see now we have this is same chamber, same particles, but I'm separating these based on the D10 and D90. See, D10 has longer residence time in the chamber. So there are smaller particles, they are fluffier, lighter, and they kind of recirculate inside the chamber, which is you can see on the left side. And on the right side, this is D90, heavier particles, shorter residence time, and they fly out faster. Now we have the mass transfer and the heat transfer, or the transfer flux, which is when we have a smaller particle, the surface area to the bulk ratio is larger. So we would dry faster, and and the particle is now exposed to higher temperature for a longer time. So the D10, if you have any decomposition or degradation happen in the D10 particles, because it's a reaction kinetic, you know, higher temperature, longer time, and we are forming like impurity in the D10 versus in the D90, longer diffusion length in the particles, they stay wetter. And when we, you know, if you're measuring the particle size and the degradation and the residual solvent content, the profile in the D10 to D50 to 90 would be very, very different. And all of this would be predictable by the models, and you can see in these examples. And then we can start running some what-if scenario. What happens if I change my temperature? Next slide, please. Yes. Now, in these kind of parametric studies, we start doing these kind of what-if scenarios. What happens if I increase the temperature or the flow rate or the flow rate of the gas or the flow or the feed flow, and what will the impact on the residual solvent content or the temperature? So now you can see we can run like multiple simulations in a very quick time and predict and understand what happened at the different scales and at different kind of CPPs, right? Combination of all of these cases now going to construct the design space. These are just two examples. We can run like multiple different scenarios, even something on like the condenser, which is like an auxiliary unit for the chamber, but it's going to change the residual solvent in the gas that goes back to the chamber and change the mass transfer gradient that's going to help us with the evaporation. So combination of all of this goes to the CMC because we are developing the design space and the PAR and NOR and also the risk analysis that if I go to lower temperature, if something fails, now we end up with some, let's say, high residual solvent content that's going to be kind of rejected product. Next slide, please.
Yeah, just want to interject here. So I think it's very important as Nima's walking you through these different pieces. What we're doing is we're building these things mechanistically. So these are based on classical thermodynamics, molecular dynamics, computerized flow simulation. And because we're doing it this way, we're basically aggregating all of the narrative that we're going to need for our module 3.2S, 3.2P. That we're going to need in our final package. So knowingly or unknowingly, as you're going through this exercise, you are compressing the time it takes for us to write that sort of story. It's incumbent upon us as product developers to build a narrative that takes us from that preclinical, pre-IND safety study formulation, whatever that was, all the way to our commercial product. We have to bridge that. That's the only way to put together an effective NDA or BLA at this point. And so by going through this sort of thought process, you're beginning to build a context around what you do and don't know, how we're going to control it. Nima mentioned PAR and NOR. There's a number of different ways to attack a PAR and NOR strategy. This allows us to run the what if. This allows us to understand what the implications would be, because at the end of the day, once we define this process, we still have our phase 3 process validation. We still have to put in place some sort of CPV framework that ultimately we have to defend and report on. So you begin to see how all of these parts, which are in the FDA guidance out there, begin to kind of coalesce. And in silico requires and addresses that narrative from beginning to end. And so we're putting ourselves in a position to answer that and describe that in our filing, just going through this exercise.
Exactly. And that follows the notion of the QBD, quality by design.
Yeah.
The quality by design is not DOE, is not just PAT, is not just showing the data, is the understanding of the correlation of the CMA, CPP, CQA. Why that happened, right? If you change your RPM, the impurity went up. Why? Right, and that's why FDA is expecting the companies to explain and understand and demonstrate that they understand the risk, they understand the process, they understand the material, and all of these mechanistic models go to like the core understanding of the correlation of different properties, parameters, and the reactions and the kinetic and all of that could also help with the review time.
Right, and so as we go through these exercises. Nima mentioned context of use, so we're going to define the application that we're using here. And what the FDA has said, EMA has said, is that we allow you to do your modeling as you're going to use your modeling, depending on what its intended use is there, but you ultimately have to validate it. And the validation is always done with empirical data. We know at the end of the day, we have to do PPQ lots, we have to do registration lots. What we're doing is we're getting rid of these intermediate experimental runs that we had to do in the past to sort of begin to understand the scale dependent, scale independent parameters as we moved up along the way. And it's allowing us to lower the regulatory risk because we're describing a relationship mathematically in silico to complement your empirical data. It's always calibrated or verified and then ultimately validated with empirical data. But that gives you huge advantage at the end of the day, and I'll show you that. Why don't we walk into the second scale up example here? Yeah.
Thanks. Yeah. So bioreactor scale up, that's another kind of very large case study. If you are interested to go to all the details, we have a separate webinar. Is a full 1 hour webinar. You can go to a YouTube channel and watch that webinar. There's also a publication on this. One of our clients and the third party, they have their even economical data also for the validation, how much saving they had. This project that I'm going to show, the client, they calculated that they had $20 million saving based on they using these kind of models for the scale up and optimization of the process. Again, going back to like the scale dependencies and what property is going to be changed when we go to the manufacturing scale. Like for example, like RPM, you can run like 100 or 200 RPM in the lab, but when we go to manufacturing, it rarely go above 60 RPM because of all the shaking and the problem that would have in the larger scale. Same for volume now, it's going to be like from 0.1 liter or 1 liter to 20,000 in the pharma, or I had a food industry project that they go to like 200,000 liters. But the gas rate, like the volumetric flow rate of the gas that goes to the reactor now would be like 100. You can see there's a huge nonlinearity for different parameters. And when we scale up that, how I should select a new RPM, how I should select the new VVM. Next slide, please. And that starts with understanding of, again, the correlation of different properties. We go to like underlying physics and the thermodynamics at each interface. We have a sparger that developed this kind of gas bubbles. This is an example for the KLA, would be similar for like a nutrients or the byproduct or the cell behavior. You can see on the blue kind of interface on the left side is a gas liquid mass transfer interface, and there are lots of properties or the process parameters that gonna impact that kind of mass transfer flux. What would be the impact of the size of the sparger? What kind of the size of the bubble that I'm forming? When the bubbles hitting the impeller, they're gonna break down, but when they go up, they're gonna coalesce. What's the impact of the pH of the medium or the flow rate of the gas, or all of these things that gonna develop the models, plural, for that level. Same approach for the inside the liquid, and then also at the liquid and the solid interface, which would be cell, or it would be very also similar for like a hydrogenation reaction. When you have like a solid instead of cell, now you have catalyst. It would be kind of same notion. Now we develop all these models, put together, run at the same time. That's a simulation, and we get the results for the KLA or the shear rate, or what's the impact of shear rate on the protein aggregation or damaging to the cells or to the product. Next slide, please. Next, yeah. This case study, we had a scale up from a very small reactor, 1 liter reactor, or sorry, 0.1 liter reactor to 20,000 liter. The large reactor at the manufacturing scale existed, so we didn't want to have any capital cost to buy a new reactor or buy new components for the reactor. But 3 different properties that we can change with no cost is like optimizing the RPM of the bioreactor, the VVM, and the orientation of the mixing elements. We could have like a Russian turbine at the bottom, the elephant here at the middle, or the other way around, because these mixing elements are like screwed on. So the operator, they could just lose the screw, bring it lower, higher, and there wouldn't be any cost in terms of the capital expenditure. So that's kind of like the constraint for the optimization, that what I can change and what I can achieve is the goal. Right? Next slide, please. So the workflow starts from like a lab scale, what properties that I can predict from the lab scale. For example, if we have some, let's say, constraint or amount of shear on the proteins that's gonna cause degradation or aggregation, these are scale independent, right? That could be like get from the lab scale very quickly. The cell behavior, the cell cycle, the cell biology, all of those things would get from the lab scale. Then we develop models and the simulation for the smaller scale. We can afford, the clients could afford to run the lab scale for the validation. Now we have validated simulations and the models that could be used now for larger scale manufacturing. Right now at the manufacturing, we use this kind of validated model that the physics and the models constraints are validated. Now we go for like a scale up, and now we start doing these kind of what if scenarios, and what I want to optimize or reduce or maximize for different reasons. Next slide, please. See, these dots are these kind of parametric studies, similar to like the spread drawing that I showed with like the lines. many different parameters, and the correlation would come from the model. In this one single example that we are showing here is the correlation of the KLA versus VVM for different impellers. These are like orientation of the impellers, or Russian turbine, or elephant ear, or a mix of both. And the size of the circuits are different RPMs. We can develop like the theoretical design space, go to like 1000 RPM. Yes, theoretically possible. It gives you higher KLA versus lower shear, or whatever correlation we can develop. But there are some constraints that comes from the physics and the economy and the material properties. Now we put this constraint, like the green constraint is when we go to high shear, at very high RPM, now we start damaging the cells. Or when we go to high VVM, now it costs more for like the recirculation of the gas, or these are some like the technical analysis that we have to develop. Now this constraint around the theoretical design space give us like the physical design space that could be useful for the controller space development, and also optimizing the process, and also the regulatory filing for the CMC. Next slide, please.
Yep. Okay, good.
Sure. This is what happened on my side, on like the background and how we run the models. I just wanted to show you like these kind of dashboards. On the top left, now we are running different designs. This is 250 designs that we run within 3 days. This is very large reactor, 20,000 liter bioreactor, and each point gives us or provide the model for like different RPM and VVM and the orientation of the buffers. We can see when we select the new points, the design on the lower right would be changing. So each design and each point are kind of correlated. And on the top right, we can have like multiple different dashboard. Here, I'm just showing the VVM versus KLA. That shows us, where these kind of points are getting kind of located. The lower left is the response factors from the models and the different parameters. Now we are running 200 different designs. We're collecting a lot of data. And from this point, we can go to digital twin, we can go to the machine learning, we can go to the surrogate models, right? When we have this data, we don't need to run the CFDs anymore, because we have studied the entire design space. Now we have a very good knowledge about the correlation of the different properties. And now we can go for like some surrogate models and the machine learning that would be very fast and, the time saving for future applications.
Yeah, so I think this is a really good illustration of what the power of this approach is. Obviously, to look at multiple different response factors in an orthogonal way, meaning in a statistically unbiased way, we're not biasing the process in any way, would require a tremendous number of experiments in order to do that. So we can do that in silico, because we've built basically a digital version of what your process unit operation is. We have incorporated the material properties that have to be considered. We've got the scale dependent factors already built in based on basic engineering and thermodynamics, and it allows us now to, with a high level of confidence, predict whatever we're interested in as an output. So all of these response factors are multiple outputs. That would be extraordinarily difficult to do empirically, right? So you can see how you take your laboratory scale data, even if you have a scaled down model for a biologic, with that, you can now run your in silico as complementary, now as a surrogate, and really expand upon that. And you can calibrate and verify against what you've done so far in terms of empirical, right? And so the beauty of this is that in a matter of weeks, we've got this snapshot here versus months, but sometimes years, to run a handful of these experiments to go forward. And so hopefully you're beginning to get a sense for how this acceleration is taking place. You can also see why regulators love this, because I'm still going to give them the lab scale and potentially scale down model. Maybe I'm going to do that as part of my normal process development. And then I'm going to add this to it. And now the regulatory risk goes to zero, because they really understand where the missing data, how it behaves in the process is. And we're going to ultimately confirm that with empirical data at scale.
Correct. As Vikash mentioned, this is more like an enabling technology. You have 250 experiments, and fermentation takes like 2 weeks, 3 weeks.
Right.
250 experiments for like 2 weeks, it would take like 4 years to done that, and
Right.
it's kind of impossible. Even beside the cost of the media. So the outcome of that model, now we can see here, we can go for like a different optimization, finding the correlation or like a threshold between the OTR and OUR, where I'm starting to starve my cells, where I'm starting to damage my cells, where I'm starting the damaging the product. And all of this gives us the kind of the best design that is, based on the models and the insight that is developed from the models that gives us, that kind of design space. So you notice, through the kind of the workflow that I kind of explained, we start from like a data to information to knowledge and then the value, right? So the modeling is a tool. Is a tool to bring the product to market faster. So that is like the business value, or providing the product for the patients is in a shorter time. So the modeling is not a goal. It's like is helping the business and the patients and the regulatory to develop this product in a cheaper and a faster way. Right. Next slide, please. We don't have much time. I go for a very quick on the next slide. Yep. Here is a oral solid dose stability prediction. Now based on the ICHQ and also the regulatory requirements, so companies, they need to do like a stability testing for 2 years or 3 years, depending on type of the product. Some we can go for like accelerated testing for like higher temperature and higher moisture or humidity, but again, they need to provide the data for like over the 2 years, and this is hugely costly. Not for the materials, for the time. What is the value of 1 day of the product in the market? For the blockbusters is $1 million per day. So now if a company could bring the product to market faster, every day they are making $1 million extra because they use models and they could do this kind of time saving. That's an example for like a temporal optimization or like in a scaling in the time.
Yeah.
Now, when we think about like kind of different degradation mechanisms, is it going to be amorphous dispersion that you might have some crystallization, or if we have like a crystalline material that would have like a polymorph change based on temperature or the plasticizer, which here is the absorption of the moisture, or it could be like any other degradation, for example, reaction with the oxygen, lights, different kind of the energy sources that could start the reaction kinetic. Next slide, please.
Well, I think the key point in this particular case study to remember is that in this particular example, there was a significant change in a late stage study to the formulation. And you can see the formulation changes here, and there was no real-time data there. And so we built a predictive stability model here and went to the agency showing that the model demonstrated that this could be used over a 30-month period. We had very limited real-time data there, and the agency accepted it because of the predictive model being built from first principles and being able to demonstrate and calibrate with the empirical data we had so far. So imagine you change your formulation in phase 2. Now you've still got a bridge between your phase 1 and your phase 3 ultimately. Predictive stability is one way for us to do that.
Yeah, and FDA highly supportive for this stability testing. They have a program called MARS, M-A-R-S. We have submitted many models to them, and they very have very good understanding about how this model should be developed and should be tested and validated, and what would the results that would be helpful for the stabilities. Here is an example. You can see like the reaction kinetic that we are developing is based on like Arrhenius reaction kinetic. We have like the NE, or like activation energy, which is like a temperature dependent. What happens if I go to 40 degrees C or 60 degrees C, and the relative humidity, which gives us the vapor pressure of the water, or the amount of water absorbed into the solid forms. How much of that water would go to the excipient? How much would go to the API? What would the interaction of this plasticizer or moisture water that with the excipient or the API, and that kind of a matrix of different reaction that could happen there. So all predicted based on this kind of reaction kinetic modeling, and now we can predict that what happened after 10 months, 20 months, 30 months at different temperature and relative humidity. As I mentioned, this is one example for temperature and humidity. We could do the other reactions like oxygen or light or different kind of mechanism of the reactions. Next slide, please.
Yep. So hopefully we're wrapping up here. Hopefully we'll take some of your questions. You're beginning to see the advantage of in silico versus classical trial and error around that. You don't have the incomplete data insight that you have typically with trial and error. I mean, you can run small scale DOEs and gain some insight there, but as we scale up, we're usually not allowed to do a lot of studies associated with it. Biologics has a lot of unit operations. That's an even more complex balancing act on how we can get to insight at the scale we're going to ultimately commercially launch this process and put together a defensible CMC strategy with this. If you marry that experimental approach with an in silico approach, you end up with a complete picture. You need some small scale data ultimately from which to understand the product specific attributes of a process. But beyond that, we don't need much in order to begin to build a clear picture of what the scale up parameters need to be. And the beauty of this is that there is efficiency that we gain as we do that, not just time efficiency, but as we move to that final process design space for your commercial process there, we don't need to run our 3 validation lots sequentially. We can run them in parallel. And so that means we end up with huge time savings around this. So on average, we have seen 30 to 40% consistently, but it's not unusual to see greater than 60% time savings, greater 60% cost savings associated with coming up with a final process design space and control strategy, which is defensible with robust sort of parameters and underlying science, which is backing up your final arguments in module 3.2S and 3.2P. And I use this one just to kind of give you a traditional path. This is like a classical biologics drug development process at the top there. You can see it takes roughly 28 months to go from this early process design at small scale to sort of your commercial lots, your 3 PPQ lots or more. Biologics, you're required to do your process validation at the time of BLA with there. So small molecule, we have the luxury of just filing it and doing it after the PAI. But if you start to look at these things, you can see how marrying in silico to that can dramatically compress. We're compressing the timeline because we're gaining insight faster here. So module 3 is a byproduct. As I mentioned to you several times during this webinar, the end game is to have a very clear and complete CMC strategy that you can defend to the agency. To us, that's module 3 there. And so in order for us to answer the questions, we're asking the questions and answering the questions in silico that we have to do in our module 3. So that's a byproduct of this exercise here. You end up with a design strategy here, a design space and a control strategy here, which fundamentally is validated because we've built a predictive model in silico and we've verified it and validated it with your empirical data there. So we end up with a robust picture of what the control strategy could look like. Nima's mentioned the PAR and NOR around this. Many ways to apply a PAR and NOR. All have implications on final yield loss, process throughput, sampling strategies, all of these things that are critical to a defensible module 3. That can be just is a typical byproduct of this exercise there. And then the one I just quickly mentioned the other day was this notion of running PPQ lots quickly. So instead of running them sequentially, I can run them in parallel with a high level of immunity because we basically understand the process design space. We know where the edge of failure is. We know what the contribution of the CMAs are and the CPPs are to the movement of the CQAs that we're interested in. So when we build our PPQ lots at scale, they're going to work. So if I'm tech transferring to a CDMO, it's going to work first time. And that's hugely advantageous not just from a cost perspective, but from a time perspective, to put you in a position to file faster. So this is our last slide here. Very quickly, we can take some questions. In silico has moved away from this innovative, academic, early R&D sort of domain to one which has now become a core component of modern CMC today. It provides you insight that regulators are going to want to see, whether it's a advanced modality like a gene therapy product or an mRNA platform or one of these advanced modalities there, or if it's classical small molecule in which you're doing scale up there. The insight and speed and confidence you can build from in silico will put you in a much better position to have a robust CMC argument for your module 3. With that, we can move to questions then.
Thank you, Bikash and Nima. Using the Conference IO software, please submit your questions through the social Q&A interface. You will also have the option to add a question by clicking the arrow next to the question.
Okay.
Can you see my screen?
Yes.
Yes.
So we can start answering some of these questions on the right here. For a regulator, how easy will it be for them to identify a poorly done model or inadequate digital twin used to support any CMC processes? Nima, you wanna maybe take the first part of that, and I can chime in. I mean, to me, a regulator, the FDA has established a 7-step model, which we have to abide by. So that's the beauty of it. They have given us a framework which is accepted by FDA and EMA on how we do our modeling. And so we have to, our reports all reflect that, the context of use, what it's going to be used for, how we went about applying our first principles, how we went about verifying and calibrating the model, how we went about validating the model. And so if that's incomplete, they're going to immediately know that you didn't follow the appropriate models there, and then you're going to have a problem. So it's fairly straightforward.
And we didn't have time for our introduction. I used to work for FDA before starting my company Procigens, and was leading this kind of new efforts for model-based process product development, continuous manufacturing, different kind of advanced kind of way of making medicine through the emerging technology team. So FDA is looking for, as Bikash mentioned, so we need to have like verification and validation. We need to define the context of use. We need to define what parameter is important and how we calibrated the models and the parameters. It's Bikash mentioned the 7-step. This is called modeling governance. Is not just how you develop the model, the documentation, the model update. If you are using the model for like a process control, now it would become like a high impact model versus low impact model for the process development. That would go to like CPV or the continued process verification for the model, for the model maintenance. So answering your question, depending on what kind of the model you are developing and what will be the application of the model. And over the past 6 years that I'm doing these kind of services for different companies, our clients submitted 6 models to FDA. So we submitted models to FDA with the results and what's the correlation of the different parameters, and it helps a lot even with the shorter review time. Right?
Yeah.
So you don't think that FDA doesn't understand model. They have very good understanding about what are the correlation of these things, what is the sensitivity of different parameters, and they know how to ask right questions.
Yeah, and so as Nima said, we've done 6 actual model filings with the FDA, more than 80 projects that have applied in silico as part of the CMC strategy, because it's up to you how much you highlight the in silico. The bottom line is the insight you gain from the model can be transparent to the agency in terms of your conclusions. It's going to ultimately define your process design space, your CPPs, your CMAs, and your final PAR and NOR, with that. If you want to lead, for example, the case study where the client changed the formulation in phase 2 and they wanted to start the study, we had to use the model as the lead in that particular case, and the FDA accepted it. So I think the future, this second question, what is the future of these models? Can they move from being, can they move further from just being complementary? 100% they can move from just being complementary. They've already done that.
We already did,
today.
yeah.
Already have done that. We had one project where the API was so expensive that we weren't really allowed to do any scale models at all. So the filing was 100% built on in silico, and the agency accepted it. So I think the confidence FDA and EMA have in modeling, the tools that are being used for modeling, the software that's being used for modeling, the governance framework, as Nima has articulated to you here, these things together give regulators confidence that the conclusions are valid and they can rely upon them. So ironically, it reduces their regulatory risk in assessing your position on that. And so you'll find that a lot of large biotech and large pharma now have implemented in silico modeling as a core step before they move to commercial, because whether you're using a CDMO or you're going to your own vertical manufacturing floor, with that, you still have some sort of a scale problem and a transfer problem that you need to do. And by doing it in silico and taking the few weeks it takes, 10, 12 weeks to run these models here, you are in a much better position to know that it's going to work the first time there. And so you end up with a much higher level of confidence that the process will work correctly. That same confidence you have is what the regulators are getting when they look at your module 3.
Correct. So it's, depending on, again, for the applications, I had a presentation a month ago at IFPAC that the model or the digital twin controls the entire process, right? So the quality of the final product depends on what the decision that is being made by the model. Is like a kind of autopilot process is model predictive control, right? See, if the temperature went up, how we can change the other parameter to stay on the CQA. For the drug device combination for devices is like highly applied, and I can give you one example. This is for like 6-7 years ago. Was for the artificial valve, and the company, they developed a new valve that is safe for inside the MRI, right? So everything was just submitted by the model, and model was used for the designing the product, risk analysis, and the validation of the product. If they want to do like a clinical trial, they had to put a valve in a patient, put an MRI, maybe the patient dead. Oops, right? So it's like the model was like the only thing that was submitted for FDA for making a decision for the review of that device.